Hello everyone, in this lecture 3 of Physiology, we'll talk about blood groups. Before we begin, here is a real story that happened in the UK. There was a woman who was 53-year-old with blood type O and a history of hepatitis. So she was admitted to the hospital due to heart failure and because of anemia as well. So following a blood transfusion, she complained about severe back pain, which decreased when the transfusion was stopped. But they continued afterwards. And when the transfusion was finished, the patient had decreased urine output and some funny urine color, which was described as tea color. She also had increased systolic pressure and also a high fever. So regardless of the symptoms, a second pack of the red blood cells was being transfused to the patient. But to their surprise, it was found that both blood packs were of blood type A, while the patient was having blood type O, and the patient died 5 days afterwards. So this was how the blood sample should look like. You could see how these red blood cells clump together because of blood coagulation. So in 2014, there were 7 cases of incompatible blood transfusion in the UK. While I do not have the data for Malaysia, but I think it also happens here, as human mistakes are everywhere in the world. Okay, by the end of this session, you should be familiar with number 1, the physiological principles of blood groups, which are the biochemistry behind it. Number 2, we'll look at 2 major blood group systems, which are the ABO system, and number 3, the rhesus group system. And you also need to appreciate the clinical relevance of different blood groups during blood transfusion. And last but not least, we'll also take a quick look on other blood group systems. And here's a bit of history on the blood group, especially on the ABO blood group system. Karl Landsteiner was a biologist from University of Vienna. So he discovered not just about the ABO blood group system, but also the rhesus factor system, enabling us to transfuse blood safely and with confidence. So he originally named the blood group system as A, B, and C, but later renamed it into A, B, and O since blood group C has no antigen. So O stands for ohne, which is in German means without, while the AB group was discovered a year later. So he received a Nobel Prize for his great discovery on the blood group system. So what are the blood groups? Well, a blood group is a classification system of different blood type based on the presence or the absence of antigenic substances found on the surface of the red blood cell. So you have learned this concept with me during our biochemistry lecture on the carbohydrates. So when an individual is exposed to foreign bodies or foreign molecules, in this case, a different type of blood cell, your body is capable of mounting an immune response towards that foreign body, which are the red blood cells in this case, specifically towards the glycoproteins on the surface of the red blood cells. That's because glycoproteins serve as the antigen. So an antigen is a molecule that can induce an immune response, and it's actually an abbreviation for antibody generating or antibody generation. So for red blood cells, the surface antigen could be a mixture of proteins, glycoproteins, and even glycolipids. Okay, we are all very familiar to the ABO blood group system, but actually there are at least 30 different human blood group systems and over 700 surface antigens that have been described. So to perform a complete blood typing will require us to screen more than 30 blood group systems and 700 antigens. So do you think it is possible? Well, it is possible, but very time consuming. So usually only the ABO blood group system and the rhesus D factors will be screened clinically. So your blood group is inherited, so it's determined by your genes. You probably have the same blood group for your lifetime, but it may change due to infection, sometimes cancer, autoimmune disease, or even after a bone marrow transplant. So as we mentioned again and again, The ABO blood group system is determined by the glycoproteins or the sugar structures on the surface of the red blood cells. And these are the major sugars that you can find on the surface, forming the antigen on the red blood cells. As you can see here, that all blood types share a common antigen structure, as in the blood type O. For blood type A or the A antigen, you have an extra what? N-acetyl-galactosamine, while for blood type B or the B antigen, you have got an extra galactose molecule. And that common antigen structure found on the surface of blood type O has a name. It is called the H antigen. So here is a structure that you can find on the surface of the blood type O, and it can be found across all different blood groups because essentially antigen H is the precursor to make antigen A and antigen B. So the ABO system is controlled by two important genes, the ABO gene with either allele A or allele B, and the H gene that comes with the dominant or the recessive H alleles. So the H gene is a dominant gene encoding for an enzyme fucosyltransferase. So judging from the name, can you guess what is its function? Well, its function is to transfer a fucose sugar unit onto the precursor substance found on the surface of the red blood cell to form the H antigen or the H substance. So showing here, you have the precursor substance, which is again found on the surface of the red blood cell. So with the help from the enzyme fucosyltransferase, we can then add a fucose unit to form the H antigen. So it's only possible with this H antigen, then we can make blood type A and blood type B by adding extra sugar units. So for your information, this precursor substance is also known as antigen I. So first of all, we will need antigen I to make antigen H, and then it's from antigen H we can make blood type A and blood type B. So because gene H itself is a dominant gene, these are the genetic possibilities. The homozygous dominant H will make antigen H. Likewise, the heterozygous dominant H will also make the H antigen, but the homozygous recessive will not make any antigen H. So this is a very rare condition. And what will happen to these individuals? Obviously, these people with the homozygous H alleles, they do not have any functioning copy of the fucosyltransferase enzyme. And because this disease was first discovered in the city of Bombay of India, it is also known as the Bombay phenotype. So these are very rare condition. It is about 1 in 250,000 within the population. But in certain parts of the India, it can be much higher. So because these people, they do not have antigen H due to the lack of this enzyme, they cannot make antigen H. And subsequently, they can't also make antigen A or antigen B, although these individuals might possess the gene to make blood type A or blood type B. So there is no negative effects with these people, but just for blood transfusion, they might have some trouble to look for individuals with the same Bombay phenotype. That's because they do not have antigen H, and therefore their body will generate antibody against the antigen H, which can be found in either blood type O, blood type A, B, or blood type AB. So it is on the chromosome 9 we have the ABO gene encoding for the transferase enzymes, either the A transferase or the B transferase or nothing at all. So if you have the A transferase, then you will be able to transfer the N-acetyl-galactosamine onto the H substance, making the A substance or the antigen A. If you have the B transferase, then you can transfer a galactose onto the H substance, making the B substance. So in short, A transferase only makes A substance or blood type A. B transferase only makes B substance or blood type B. If you have both, you're going to make a blood type AB as a result. Okay, here is a diagram showing the formation of antigen A and antigen B. So for all blood types, we begin with this precursor structure, which is also known as the antigen I or the I substance. So with the H gene encoding for the enzyme fucosyl-transferase, we can then transfer a fucose molecule to this I substance, making the H substance or the H antigen, which is also the antigen on the surface of blood type O. So depending on the alleles that we have on chromosome 9, we can then make the A antigen if we have the A transferase, or the B antigen if we have the B transferase. And for certain people who possess both the A and B transferases, they can make antigen A and B, and therefore they are said to have the blood type AB. And now, given the genotype of the individuals, can you please guess their phenotype or their blood group? For example, with the homozygous AA in the first example, what you'll be getting is blood type A, because you are going to get A substance only. How about the second one? AO is going to make A substance only, or BB is going to make B substance only, BO, B substance only, AB, you have both A and B. What about blood type O? You are going to get substance H only. Again, here is a summary of the same thing. It is just some basic genetics. As long as you understood the concept, you should be able to tell the phenotype based on the genotype. So what is interesting is that we can actually use the ABO system for paternity testing or for forensic analysis. By looking at the genotypes of the parents or the matings, we can rule out the impossible children. So for example, there is no way for a couple with blood type O having any children with blood type A, B, or both. I believe you can work this out simply using your logic and reasoning. But there is a caveat to it. Certain diseases might actually change your blood type, as I mentioned earlier. It could be infection, it could be cancer, or even some form of autoimmune diseases. So just now we have discussed that the H allele itself is a dominant gene. So you have the dominant H and the recessive H alleles. But in the case of the ABO gene, both allele A and allele B are dominant. So in this case, we call them codominant alleles. So both allele A and B are codominant alleles, which means when both genes or both alleles are present, both antigens will also be expressed. So a couple with an AO genotype and a BO genotype can potentially produce offsprings with 4 different types of blood types. Could be blood type A, AB, B, and O, which is quite interesting to see. Now imagine if you are a genetic counselor and your customer came to you with this question. My husband and I have blood type O, but my kid has blood type A. How it is possible? So what would be your answer? Please pause the video for a while and ponder on this question. Well, there are at least 4 possibilities. Number 1, it is just some bad blood testing. So it could be human mistakes in performing the test or some mistakes in reporting the blood type. Number 2, it could be due to mutations or certain diseases, as we discussed just now. Or a third possibility could be 2 babies getting switched during birth accidentally. So that is again due to human mistakes. While last one, someone else could be the father. So how do you rule this one out? Well, you can carry out a genetic testing.